Okay, well, it's 1:15 now, so I'll get started. And if anybody joins, they'll join in the middle. It will turn off the notification so it doesn't bing too loudly. So yeah, welcome everybody to this week's CPS talk. We've got a really interesting talk this week on finding cures for triple negative breast cancer. And it's Professor Swen Hoelder who's giving the talk. He is a key member of the Cancer Research UK Cancer Therapeutics unit. And he applies his medicinal chemistry experience to discover and develop new cancer drugs. He's worked in really wide range of places in both academia and industry. He's worked at the University of Münster and the Technical Uni in Berlin, and as far away as Berkeley in California. He's also worked for Sanofi and Takeda in industry, and now works at the Institute of Cancer Research. So yeah, I think it'll be a really interesting talk today. So I will let you get started, Sven.
Okay, thanks for the kind introduction. So I'll try to share my screen and put it into the presenter mode. Can you see, can you just let me know, can you see the proper display of the slide, or do you see
Yeah, no, we could see the chat, but now we can just see the proper display. It's fine.
Okay, let me just move.
All good.
Okay. Okay, yeah, first of all, thank you very much again to Chiara and the Chemical and Physical Society for this kind invitation. It's a great honor and a great pleasure to give a talk here. And so I'm a medicinal chemist by background, and so the obvious thing for me to talk about is one of our drug discovery projects. So I've been at the Institute for about 13 years now, and I've worked on a variety of different drug discovery projects. But I was a little bit worried about doing that with a broader audience, with broader research interests. And I was a bit worried about putting everyone to sleep with our jargons and our abbreviations. So I thought I'd do something different. And so what I'm doing is really talking about why is it such a huge scientific challenge to discover effective cancer treatments. Now, cancer is a complex biological phenomenon, right? And I'm by background, I'm a chemist, right? So I'm doing something I really shouldn't be doing, which is talking really about something where I don't have the fundamental and deep expertise. But obviously at 13 years at the Institute, I've picked up quite a few things, talking to colleagues, reading, and so I want to share some of that, and hopefully that'll be interesting. And then in the last couple of slides, I will just whiz through one of the programs my team was involved, where we worked on inhibitors of MPS1 and progressed the compound to clinical trials. And so I just give you a really quick overview about this program to give you an example of how we work on cancer drug discovery programs. Okay, let's get started with the basics. So what is cancer? First of all, cancer is a pretty big killer. So the World Health Organization, WHO, recognizes that about 1 in 6 global deaths is attributed to cancer. So many lives are sadly lost to cancer. Another important thing that is, I think, quite important to understand and recognize about cancer, that it's not one disease, it's a collection of about 100 diseases. Cancer can affect virtually every part of the body, every human being, and lung cancer is very different from, for example, breast cancer or pancreas cancer, and will require different treatment. So sometimes I ask by people, when will there be a treatment or a cure for cancer? There may be cures eventually, but there will be certainly a lot of different cures for different cancers required, right? So of course, what all of these different diseases have in common is that they're all characterized by uncontrolled growth of cells in our bodies. So our bodies consist of about 1 trillion cells, and that's really a mind-blowing number, right? And of course, we can only function and work as a multicellular organism if all of these cells really do what their job is and really stick like a musician in an orchestra, right? And this is where things go wrong in cancer, where individual cells start to proliferate in an uncontrolled fashion. So they start to grow even though they're not supposed to grow, right? And initially, this may be not such a big problem. You have a few more cells, but generally the growth will become faster and faster in an exponential fashion. This exponential growth we all probably have heard a lot about recently, and will start to affect the function of these organs. And so eventually, as the cancer develops and becomes, if you want, more nasty and more aggressive, it will then start to really sort of damage the functions of vital organs, and it can then also spread via the blood system, via the blood vessels and lymph systems to other organs and affect other parts of the body. For example, the brain is a common place for metastasis, or the bone marrow. So what causes this sort of change in behavior to uncontrolled growth? And that's very well understood. And the simple answer is DNA mutations. So the function of each cell, so the role each cell in our body has to play, is of course hardwired into our DNA. It's if you want, if you use an analogy with computer, it's coded in our DNA. And now, an important thing to know about DNA is that it's constantly subjected to DNA mutations, right? There are different sources of mutations. Some mutations just happen naturally, right? When cells divide, there's a certain error rate that always occurs. There are, of course, other well-known sources for DNA mutations and mutations to the DNA. For example, smoking, of course, is a big source of for mutations in your lung. Radiation is well known. Sometimes it's simply bad luck, right? And you inherit mutations from your parents. Most of these DNA mutations and changes that happen all the time are completely irrelevant and don't interfere with the function of these cells at all. But certain and very rare combinations of mutations can override and relieve the growth restraint that every cell in our body has, right? And can override this restraint that the cells is not supposed to grow, right? And then causing this sort of uncontrolled growth. It's really important to say that these transforming events are very, very rare, right? And often a sequence of mutations, a specific sequence of mutations is required to transform a normal cell into a cancer cell that grows in an uncontrolled fashion. So it's very unlikely for any individual cells that will ever transform into a cancer cell. But because our body has so many cells, as I mentioned earlier, about 1 trillion cells, right? There is a significant chance that each of us is eventually diagnosed with cancer. And another thing probably worth keeping in mind is that mutations, of course, again, they constantly happen, but they also accumulate as we grow older. And so the likelihood of having the correct number of mutations that can transform a cell, of course, then increases with your age. And that is the reason why cancer is more often diagnosed and diagnosed in older ages. So cancer is a part of life, it's a matter of life. So how can we deal with cancer? How can we intervene against cancer? And really, we have, on a basic level, 3 lines of intervention. The first one is very simple, right? And doesn't need much explaining. In the first instance, prevent cancer, right? And again, the World Health Organization, if you check their webpage, reckon that about half of all cancers are caused by lifestyle and are thus, at least to some extent, avoidable. And again, I also already mentioned some of the sources for mutations in your lung or your skin that can lead to deadly cancers. Now, it is unrealistic to assume that we can prevent all cancers. So the next level of intervention is that early detection of cancer. It is sort of difficult aspect of cancer, especially of some cancers, is that they grow for quite some time completely unnoticed because they're not causing any symptoms. So a patient who has a cancer growing or tumor may not immediately recognize that this cancer is developing. And so this cancer is developing silently, and as it develops, becomes more aggressive, larger. And when it's finally diagnosed and found, it can be too late to cure it. Colon cancer is a good example. For I think colon cancer can grow for about a decade in your colon without being noticed, right? But when it's finally noticed because there are symptoms, for example, bleeding or pain, it is often very difficult to treat because the cancer has already spread and has already become quite large. And so this is a really active area of research, and also an area where a lot of progress has been made in many types of cancer to diagnose them earlier and find these cancers earlier before they have really led to symptoms, and remove them at this point before they have transformed into something really aggressive. Now, when a cancer is diagnosed, it will normally require some sort of treatment. And so treatment is really the third line of intervention against cancer. And really we have three broad classes of surgery to our disposal: surgery, radiation, and drugs. And so the prognosis for many cancers have really improved significantly in the last, I would say, decades, due to early detection and treatment. So many patients, when they're diagnosed these days, they will have a much better prognosis, a much better chance of survival, or for long-term survival, than they would have had maybe 10 or 20 years ago. But as I mentioned early on, it's still also a fact that there are many cancers where we can't do much for these patients that remain incurable. And as I mentioned also, many lives are lost to cancer. And that really raises the question, why is it such a huge scientific challenge to discover effective cancer drugs? I mean, the war against cancer, I think in the US was launched in the 60s. And since then, huge resources, lots of brains, a lot of manpower went into finding new cancer drugs. Why has that proven so difficult? Well, let's start with the challenge. What does a cancer treatment need to achieve? A cancer drug needs to selectively and quite potently kill or contain all cancer cells. You cannot miss a single cancer cell. You want to kill or at least contain all cancer cells in your body. So you need a pretty potent weapon against cancer cells. On the other hand, it needs to be very, very specific and selective, and does its job of killing cancer cells without causing lethal damage to normal tissue. And this is quite a tall order. Now that is something that surgery and radiation, of course, can do very well, right? In surgery, of course, you only take out the tumor as much as possible, and radiation, which has developed a lot also over the last 20 years and has become more and more precise, also can really focus on the tumor. But of course, not all tumors are amenable to surgery or radiation, and that's why we need drugs, and drugs that we can apply systematically. So a drug that, for example, you swallow or you receive via an intravenous gavage. Now, cancer drugs have a long history, so early chemotherapies, and these were pretty much discovered serendipitously. So someone would find, for example, a natural product and observe that in a petri dish, this would kill cancer cells, and when given to first animals, then humans, was sufficiently tolerated by this patient to have an effect and have a then positive effect on the growth of this cancer, and perhaps even cure this patient of this cancer. Now, some of these chemotherapies are still in use, and some of them are very effective. But of course, most people will be familiar with the sometimes really quite difficult and sometimes terrible side effects these chemotherapies have, right? Really leading to quite a significant drop in quality of life for these patients. And so for a long time, there was this desire to discover drugs that are kinder to the patients, that are more specific for cancer cells and spare more normal cells, right? And really the opportunity to do this became then available when, in essence, large-scale sequencing became available. I think the first complete human genome was probably sequenced about 15 or 20 years ago, and one of the many exciting applications of high-throughput sequencing was then to sequence genes of cancer patients. So take tissues from these patients, tissues from their cancer, and sequence their DNA. As I mentioned earlier, cancer is caused by mutations in the DNA. And so when you find these mutations that transform the cancer, then of course you have a tool in your hand to find drugs that specifically address this mutation in a specific manner, because this mutation would not be in a normal cell. And this was really then explored, and we often use in this context the expression of, for example, cancer dependencies, where mutation on which a cancer relies, or also the Achilles heel, right, of the cancer, because again, that's something that distinguishes it from normal cells. And so then we have, I would say, in the last 20 years or 15 years, explored these specific mutations to explore so-called precision medicines. Now, just an example here, and one of the early poster childs of these precision medicines were BRAF inhibitors and BRAF inhibitors for the treatment of malignant melanoma patients. So two things probably which need explaining here: what are BRAF inhibitors and what are malignant melanoma patients. Let's start with the melanoma. Malignant melanoma is a very nasty skin cancer. I'll show you an example in a couple of slides. And until maybe 10 years or even 5 years ago, was almost impossible to treat. So there was not much you could do for these patients, right? Now, when sequencing became available, what researchers then did was, again, isolating tissue from these melanomas, from the skin of these patients, and sequencing them. And what these sequencing efforts showed was quite a few of these patients, about half, if I remember correctly, had a mutation of the BRAF gene, right? So what is BRAF? BRAF is a kinase, right? And as many kinases, it is part of a signaling transduction pathway. So it's, if you want, an on-off switch. And here it functions together with this whole pathway to connect the growth of these cells to what's going on outside this cell. So what's happening here is that growth factor that we all have in our bloods dock on this cell and cause this cascade, activate BRAF kinase, and this is a signal for the cell to grow. The important thing, though, is that if there aren't any growth factors, and if this is not activated, and there are other ways to activate these pathways, then this pathway is essentially silent. And this BRAF kinase is an enzyme that takes a phosphate group from ATP and transfers it to other proteins. If there is no stimulus from the outside, this kinase is silent, is catalytically inactive, and doesn't activate this pathway. Now, what these researchers that did the sequencing of the melanoma tumors found was that many of them had a mutation in this BRAF gene, and this mutation would constitutively activate the kinase. So this kinase now, this mutated kinase, is constantly active, and it's no longer an on-off switch, it's constantly on. And this then leads to as one of the main drivers of this uncontrolled growth of these skin cancer cells. Now, this, of course, immediately then triggered the idea of developing inhibitors for this BRAF. It's a kinase. There are many kinase inhibitors out these days, right? And so it was a huge effort, many big pharmaceutical companies. We were also involved at the Institute of Cancer Research. And so there are at least 3 inhibitors. You can see them now here that are now approved for use in these melanoma patients which have a BRAF mutations. And vemurafenib, these kinase inhibitor names are always difficult to pronounce, was the first one to become available to patients. And these inhibitors, on the whole, proved to be effective and at least delayed the disease in many cases. And again, I will show you a bit more data later. So this whole idea of precision medicine, of targeted therapies, or therapies targeted towards a specific mutation, at least to some extent worked. But we also quickly learned that there are many challenges associated with this concept of targeted therapy and precision medicines. First of all, as more and more cancer genomes were sequenced, we also found quite a large variety of different drivers of cancer's growth, right? Meaning that these different cancers would require different drugs, right? And it's coming back to something I mentioned earlier, right? Where we probably need different drugs for these different cancers. But even what we then learned is that different lung cancers, so 2 tumor samples that look under the microscope exactly the same, may be driven by different genetic events. Again, meaning that even though they are almost identical looking tumors, they will require different treatments. So, so it became relatively quickly clear that if we want to, pursue the dream of eventually curing most of all cancers, we would need a large number of drugs, right? The other thing we learned is that some refer to these patients that share a mutation and can benefit from the same drugs. We refer to them as patient population. The other thing we learned is that some of these patient populations are very small, right? And there are some examples of maybe 50 patients per year in the UK, perhaps a few thousand patients worldwide. If you then have in mind that developing a new cancer drug and bringing it to the market often costs about £1 billion, you can see that this is economically incredibly challenging to achieve, to discover a drug for these small patient populations. Another challenge that we incurred is that despite really extensive sequencing, there were also some cancers where we simply didn't find any therapeutic points for therapeutic intervention. There were no clear mutations that would, or mutational signatures that would give us an idea of how we can target these cancers. And finally, some driver mutations were identified, but they were not amenable to drug intervention. A good example are transcription factors. There's a transcription factor, for example, referred to as MYC, and it's a very flexible protein. So there's really no binding sites for small molecule drugs. It's intracellular, so as of yet also cannot be targeted by protein drugs like antibodies. Or, for example, other driver mutations are so-called loss of function mutations. So rather activating a kinase, maybe another kinase is lost, and that's of course much harder to mend with a small molecule. So these are some of the challenges, but I think the most scary challenge to this concept of targeted therapy and precision medicine is shown here is resistance, right? I'll explain that in a few slides. Apologies, this is probably not nice to look at, but I think it really exemplifies the problem, right? And this is really coming back to these BRAF inhibitors, right? As I mentioned, they eventually became available probably about 10 years ago. This first example here, vemurafenib, right? And this was given to this patient, and this is one of these melanoma patients. You can see how this patient looks before treatment, and you can see this skin cancer lesions all over this patient, right? And then this patient receives treatment, and that's a few weeks later after start of treatment. You can see that most of these lesions have actually gone away. And for me, as a non-expert here, it's probably hard to spot that this patient ever suffered from cancer. But then, sadly, another 3 months on, the patient has sadly relapsed, and the tumor no longer responds to the cancer. And if you take the time to compare these two pictures, you will see that most of these cancers simply regrew. So it's not new lesions appearing; it's the old ones. It's the old one coming back. And again, I think this is really the most scary challenge of finding effective treatments for cancer. And we've seen this for many targeted drugs. And the clinical reality, and I think that's accepted now, is that we will find resistance for almost all new targeted drugs. So that, of course, raises the question: how can a tumor become so quickly resistant? I'm really talking about a few months, sometimes just a few weeks, to a treatment that, in the first instance, is very effective. And I tried to explain that in this cartoon, and the answer to this question was, again, a further enhancement in DNA sequencing. So when we started this initiative of sequencing cancer genes, the technology had evolved such we were able to take a lump of cancer, measure it up, and determine an average sequence for all of these cells. By the way, this is just a symbol for a cancer cell, right? So this is if you imagine this as a lump of cancer, and these as individual cancer cells, right? And so this, and of course, all of these have DNA, and you can sequence the DNA of all of these cells. But initially, this wasn't possible. And so what we achieved from sequencing was an average sequence, right? And this average sequence perhaps gave us the idea that these cells, in terms of the DNA and in terms of the mutations, are quite similar, right? And then the idea is we just design a precision medicine that binds to it, and this would then kill all of the cancer cells. Now, technology moved on, and cancer DNA sequencing moved on, and this eventually allowed us not only to determine an average sequence, so just measure all these cells up together and sequence them, but to isolate individual cells and sequence them, right? Sequence individual cells, sometimes for tumor sample, thousands of different cells, right? And they showed us that the picture is much more complicated, and there's a huge amount of what we refer to as heterogeneity. This means that all of these cells, even though they are part of the same cancer, may even sit next to each other, all have subtly different DNA, so they all have different mutations. And what is now happening, and I think that's accepted again for many therapies, is when you now treat with your drug, there's already a resistant cell there. Now, again, a numbers game. If you assume that if you look at any single cell, the likelihood that it will ever become resistant is very, very small. But again, especially large lump of cancer can have many, many cells, up to 1 billion cells. And again, you reach numbers where it's the likelihood that there is already a resistant cell is fairly high. Of course, what's happening then is that initially you kill the lump of cancer, and the cancer becomes much smaller. And that's exactly what let me just go back, observe here, and the patient looks better. But this one cell here survives, and essentially just takes up the space, and the cancer quickly regrows, right? And in regrowing, it will again become more heterogeneous and develop new mutations, right? So one of the challenges of cancer is really that it's not only really quite a lot of different diseases, but this disease never stands still, and this way can become resistant to many treatments. And again, the important point here is that the seed for this resistance is already there when we start treating, right? There's already this resistance cell there when the patient first received the drugs. And I think that's what most people know in the community really perceive, again, as the biggest challenge, scientific challenge, this aspect of heterogeneity to cancer therapy. Now, again, slightly coming back to this aspect of deep sequencing, this ability to sequence very different cells, and has provided us very different views of cancer. We're now able to establish this tree here. This was just one I was easily able to get from a paper, right, where you sequence, you take one sample, the primary tumor, and you sequence many of these cells, and then you can take different samples, metastasized samples, perhaps in a different body, but you can also take a sample of a tumor before and after treatment and compare these two cells and see which clone in the original primary tumor has outgrown to become resistant. So we can establish these trees based on the site or perhaps also on treatment. And this probably reminds you of something, and correctly so. This, of course, reminds you of these trees that Charles Darwin drew many years ago. And in fact, we really know, and that's again largely due to this advanced in sequencing, regard cancer as an evolutionary disease, right? That cancer progression, spreading and developing resistance are really revolutionary processes. And the concept of evolution is now commonly used to analyze and describe cancer, and has made a big difference. So how can we then tackle this problem of cancer evolution, of heterogeneity? And I'm afraid we don't have the answers yet. I think the community is pursuing different concepts how to tackle this problem of heterogeneity and resistance. I just sort of have a list of a few of them, and I'm sure there are others, right? And a really important point that's again pursued is to understand the evolutionary trajectories that cancer cells explore to become resistant. Now, evolution is very powerful, as we know from life, right? We are a great example of how something very simple has developed into something very complicated. But of course, evolution also works within constraints, and that's exactly what we also observe within cancers, and that cancers that harbor the same mutations, are driven by the same mutations, often also follow very similar pathways to become resistant. And so this understanding how cancers develop resistance, but also the constraints that exist for these cancers, will help us to come to more effective therapy. And again, that's a very active area of research. Another way, and that's inspired and borrowed from antivirals, is to use combination therapies. And so here the idea is there's a significant likelihood that one cell is resistant against one treatment. Yeah, because tumors can be quite large, have about 1 billion cells. There is a significant chance that one of these cells is already resistant. But the chance that there is a cell that is resistant against two mechanistically complete distinct therapies is much smaller. So at least mathematically, that is probably a game that you can win. And again, it works really well for antiviral therapies. HIV is a great example where treatments are given as combination treatments. Now, it has proven to be challenging to explore this concept of combination for cancer, and the reason is that, different from antiviral therapy or antibacterials, we're still targeting human genes here, right? And most even targeted cancer therapies have quite significant side effects. And you also have to keep in mind that cancer patients are often weak, right? And by combining two different drugs, we're also adding the side effects. And so combining two agents is not always tolerated. Nevertheless, I think this is a really important, it's an area that has shown success, and I think we will continue to explore as a cancer drug discovery community. Now, another concept is one that I already mentioned earlier, and just coming back to that, is of course early detection. As the cancer grows, it accumulates more and more mutations, and the more mutation the cancer has accumulated, the more heterogeneity it has accumulated, the better the chance for this cancer to have one that is already resistant to the treatment. And so the earlier we catch the cancer, the smaller the heterogeneity and the smaller the chance that there is already a resistant clone in there. Another one is immunotherapy. That's not something I know a lot about, but there's a lot of excitement about that. And the idea is here to essentially harness or recruit our immune system to fight off cancers, right? And the immune system is preventing us from many, many cancer cells. So we would have a lot more cancers if the immune system wouldn't protecting us from these and essentially killing these cancer cells. But cancers learn to adapt to the immune system and escape the immune system. And recently there has been a lot of exciting development in sort of unleashing the immune system, so wakening up the immune system to recognize these cancers. And some spectacular successes with drugs, antibody drugs, and cures in cases like for example, these skin cancers I showed you earlier. So it can be very, very effective for some patients, but unfortunately, they don't work so well for many other patients, right? And again, it's an area of ongoing research to understand which patients respond well to these immunotherapies, are even cured by immunotherapy, something that is almost unheard of for drugs, and which patients don't respond to perhaps, find new immunotherapies and different ways to give these drugs to make more patients benefit from these drugs. Now, another concept is more, again, seen in the evolutionary context, is to contain the cancer rather than kill it, right? And the idea is rather than, killing all these cancer cells to make room for an even nastier cancer to fill this gap, is to sort of find a way to contain it. So it doesn't grow any further, but it also doesn't go away. In essence, to find a way that patients can live with their cancers but won't die from it, right? Again, something I don't know a whole lot about it, but I think certainly intellectually quite an appealing concept. And a final concept I want to mention here, because I will come back to that, is going, if you want, exactly the other way around, right? Is going back to drugs that are a lot more aggressive, perhaps less kind to the patients. And the idea is here, by developing really highly specific, highly precise drugs, we've made the cancer cells very easy to get around them, develop resistance. Perhaps we need to go back to this idea of stronger chemotherapies, that some of them again are effective, and that it's not that simple for cancer cells to get resistance or develop resistance against. We can just see how we're doing with the time. Okay. So now going to switch topic for the last 10 or 15 minutes and talk about one of the drug discovery projects that has been going on at the Institute of Cancer Research, in which my team was involved for several years. We start by introducing really where I work, the Institute of Cancer Research. The ICR is an independent research college at the University of London, and we have two sites, one in Chelsea and one in Sutton. And all the chemists are based in Sutton. And if you check our webpage, you will see that our mission statement is that at the Institute of Cancer Research, our mission is to make the discoveries that defeat cancer. And there's a bit of both going on, or quite a bit of both going on. First of all, there's a lot of basic research going on, trying to understand how cancer works, what causes cancer. But then the Institute of Cancer Research, and that's perhaps different from many other academic institutes, has a large emphasis also in what we call translation. So translating these basic research findings from the institute and other places into patient benefit. And that patient benefit, this translation, occurs at several levels at the institute. For example, clinical trial designs, right? Clinical research is a very, very important part of cancer research, of finding new drugs. And it's quite a complex matter, and a lot of research is going into these conducting clinical trials in an effective manner at the institute. But there's also large emphasis on physics, for example, new radiation technologies. Imaging is a big topic here. It's very important to be able to image a cancer inside a patient. And then, of course, we also have a large emphasis, and I'm part of that, on drugs, and especially small molecule drugs. So I'm part really at the CRUK Cancer Therapeutics unit at the Institute of Cancer Research. So what is that? What is the CTU? So we really see ourselves as a drug discovery arm of the ICR. We are focused on small molecules. We are about 150 scientists, and we're actually just about to move into a new building here in our Sutton campus. We're really excited about this. So you could say we're in many ways structured like a mature biotech company, but under an academic roof, right? One of the great advantages we have is that not only we, but also the Hoelder Institute of Cancer Research, is that we have close links to Royal Marsden Hospital. Royal Marsden Hospital is one of the leading cancer hospitals, I would say, in Europe, if not worldwide. And many people who have appointments at the institute also have appointments at the hospital and treat patients there. And that's hugely important for discovering cancer drugs. So within the CTU, we run a portfolio with this 150 people of about 8 drug discovery projects. We have several compounds in clinical trial, and we are very proud that one of our projects has led to a marketed product. Abiraterone was completely designed and synthesized at the ICR, I should say, long before I joined, and developed at the Royal Marsden Hospital and was approved in 2011 and really changed the course of treatment for many patients with advanced prostate cancer. So as I mentioned, we have 8 projects. My team is normally involved in, I would say, 2 to 3 of them. And again, because we're already quite fine to the time, 30 minutes now, I'll make it short and just focus on a few slides. So the example I want to show here is MPS1 inhibitors as a treatment of triple-negative breast cancer. And again, there are 2 things probably need explaining here. What is MPS1 and what is triple-negative breast cancer? Now, MPS1 is a protein kinase again. As I mentioned earlier, protein kinases take a molecule of phosphate from ATP and transfer it to often another protein. And doing that, they act as a switch, an on-off switch. And I'll show you a bit more of the biological process that MPS1 is involved in. So what is triple-negative breast cancer? Well, triple-negative breast cancer is a fraction of all breast cancers. It accounts for approximately about 15% of all breast cancers. It's often diagnosed in younger women. One thing that is specific about breast cancer is maybe a bit difficult to explain in this audience here is, but it's one of these populations where we haven't found driver mutations, with a few exceptions. But many cancers, despite sequencing triple-negative breast cancers, there's no clear driver identified, and that's different from many other breast cancers, where, for example, the estrogen receptor or HER2 receptor drive the disease and can be targeted by small molecule. As such, it's such a quite heterogeneous group again, and it's very challenging to identify new targeted therapies. The normal treatment for patients is chemotherapy, often in combination with surgery. This initially works well, but sadly it has quite a high rate of relapse, with a peak recurrence after 3 years post-surgery. The positive thing is if patients have achieved or haven't experienced relapse after, let's say, 5 years, often they're then really considered cured. Nevertheless, because of this high relapse rate, triple-negative breast cancer is considered a cancer of high unmet medical need. And again, I can't go into the detail, but my colleague at the institute here, my former colleague, Professor Spiros Levanadopoulos, he identified through a series of really elegant and interesting biological experiments that MPS1 is a good target for triple-negative breast cancer. So what does MPS1 do? And that's, again, a bit difficult to explain, so please bear with me. So MPS1 plays a big role in the cell cycle. So what is the cell cycle? I assume most of you are familiar with that, but essentially, growth and proliferation is all about cells dividing. So one cell divides into 2 cells. And this process of dividing is generally represented by this so-called cell cycle, and has 2, in a very natural, very simple level, it has 2 key stages. In the first stage, this mother cell duplicates its DNA. So of course it needs 2 sets of chromosomes, one for each of the daughter cells. So this is often referred to as the S phase. And then in the second stage, what this cell now needs to do is to separate these 2 sets of chromosomes and move them to different ends, so that this large cell can now divide into two cells. And this separation of the DNA is really quite a feast for the cell to achieve, and it's achieved through this mitotic spindle. And I'm sure you've seen pictures of this mitotic spindle. So what happens here is that all the chromosomes are lined up along a plate, and one set of chromosomes for one daughter cell is attached to one pole of the spindle, and the other one to the other part of the spindle. And once everything is lined up and connected through microtubule, these are then through shear force pulled in opposite directions. Now, an important thing to know about mitosis and this mitotic spindle here is that it really needs to work with high precision. For example, if this pulling starts before everything is correctly attached through microtubules, the DNA won't be distributed. And one of the daughter cells will have more DNA, and the others will have less chromosomes. And this will typically be deadly for these cells. And so cells have evolved a number of mechanisms to assure that the cell cycle, especially mitosis, occurs with high precision. And MPS 1 is now one part of this. So essentially, when MPS 1 is catalytically active and phosphorylating, this means do not go ahead, do not start this act of pulling. And once the mitotic spindle is properly formed, completely formed, MPS 1 is silenced and deactivated. And the actual act of mitosis and pulling these two sets of chromosomes and DNA apart can actually start. So, as I mentioned, Spiros had identified MPS 1 as a promising target in triple-negative breast cancer. How do we rationalize that MPS 1 inhibitor is such an important, checkpoint? Why would this be effective in triple-negative breast cancer? So, what happens if we inhibit MPS 1? Well, if we inhibit MPS 1, we override this checkpoint, and this means we will have, this will maybe not necessarily this, but we will have an increased number of cell divisions before the mitotic spindle is correctly formed, right? And again, this will generally lead to cell death. Now, cell death is something, of course, that we like for cancer cells. We want to kill cancer cells, but it's important also to recognize that many normal cells on which we rely in our bodies, for example, blood cells, go through this cycle all the time. And of course, these will be affected as well. But the hypothesis that we explored, and based on Spiros data, is that triple-negative breast cancer cells, because they have a lot of aberrations already in their mitotic machinery, right? So this whole process in triple-negative cancer cells is often already somewhat clunky through going through the proliferation so quickly, through accumulating mutations, that they highly rely on MPS 1 to be able to come up with or establish a functioning mitotic spindle at all. And so the hypothesis that we explored here was then that by inhibiting MPS 1, we have a stronger effect on these triple-negative breast cancer cells than normal cells, and thus hopefully an effective therapy. So with this hypothesis, we then went ahead and started the drug discovery program. And I just said one slide on this. So normally 20 or 25 minutes of my talk would be on this whole process here, and I condensed all of that into one slide to be able to fit it into the format of this talk. So just quickly take you through it. We started this project with a high throughput screening. I guess most of you will be familiar with that, but what we do here is really take quite a large number of compounds, often 200,000 compounds or maybe 1 million compounds, and all screen them against MPS 1 to identify inhibitors. And the result of this high throughput screening was this was the Caesar-Indol compound here, and was an excellent screening hit, even though it had several problems that we needed to work on as a medicinal chemist. So for example, it was a very unspecific inhibitor which inhibited several other kinases. But again, that's normally the situation. That's our job as medicinal chemistry, to work on this, make more molecules, and subsequently improve the properties of this compound. And so the first step was then really to take this forward. We made perhaps 100, 150 compounds, and then helped by crystal structures, discover this highly specific important compound. So this is about 10,000-fold more potent than the early compound. It also only inhibits MPS 1 out of the 500 kinases in the human kinome. So a great drug. It was a great tool also for us to look at the role of MPS 1 using inhibitors. But there was one problem that we really couldn't sort out. And that was, and again, I can't really go into detail, was that this drug had a high risk of inducing drug-drug interaction. So in essence, if this compound is given together with other drugs, there is a risk that because this drug is here, other drugs may have higher concentration. And if you think about chemotherapy, again, this could be deadly. And so after putting quite a bit of work onto here, it sort of dawned on us that this was a dead end. We couldn't take this forward, right? Quite a difficult moment in the project. It's not a common, again, in medicinal chemistry, and sometimes the analogy that is used here is the Rubik's Cube. I guess you're probably familiar with the Rubik's Cube, and the analogy is that It's not that hard maybe to get 3 or 4 sites correct, but to get all of them correct is really the big problem, because you may have 3 and then you try to get the 4th site, and whilst you're doing this, you mess up the 3rd site or the 2nd. And that's exactly the problem we face in medicinal chemistry. We try to work on one problem and make it better. But in doing that, we have to do changes to the molecule, and these changes may introduce other problems. So this really turned out to be a dead end then. And then we followed a strategy that is, again, quite commonly explored in this situation. It's called rescaffolding, because we couldn't take this structure class forward. We used our structure-based knowledge and our knowledge of the structure-activity relationship to discover a new series of MPS I inhibitors, these pyrrolopyrimidines. Not pyrrolopyrimidines, pyridopyrimidines. And this turned out to be a very productive avenue. And you can see a second lead structure here. So perhaps another 150 to 200 compounds going from here to there. And again, we had a compound in hand that was highly potent, highly selective, just like this one. It was a bit smaller, so had better drug-like properties. But there was, again, one thing we really struggled with optimizing. That was, in this case, there was a huge risk that this compound would be, or this drug would be, metabolized in human livers. So there are simple test models for that, essentially liver cells, where you can test for that risk. And this was not acceptable. So this compound we couldn't take forward as a drug. But we essentially found a way around, and the key to that problem was essentially this methyl group. We once introduced this methyl group, and we really discovered that by serendipity, the compounds become completely stable in human livers, at least as judged by these models. And this really led to this discovery of this compound here, this drug BOS 172722, that is currently tested at patients in phase 1 clinical trials. Yeah, probably leave it there. Just finish with one slide, because this is a chemistry society. So just finish with one slide of chemistry to show some chemistry after all, and show you how we make this compound, how we made up to 1 kilogram, not we, but the CRO made up to 1 kilogram of this compound. So we made basically all of these compounds. You can see there's a pyrimidine and there's a pyridine in this compound. And the quite effective route forward that we found, it wasn't that difficult, was to start with these pyrimidines, where we have a carboxylic acid here and a halogen here, and then introduce this unit here to close the pyridine. And that worked really, really well initially for the compounds without this methyl group here. So just to introduce these 2 carbons was straightforward. But where we really encountered a problem was with this methyl group, and now we need to introduce, of course, these 3 carbons here, right? And we use different palladium catalyzed reactions, Stiller couplings, Heck reactions, Sonogashira couplings, and they all really didn't work. And palladium is not great for upscaling drugs, because it's very toxic, and there are very, very, very low levels of palladium allowed in final drugs, batches of drug. And so that was a big problem, because we needed more drug for our animal trials, but also then for the patients, and struggled to make it. Finally, a postdoc in my team, Paolo, he had this idea and saw this reaction, published actually for a very different system, right? Very different system from our pyrimidines. And it's quite a counterintuitive reaction, where you take this kappa catalyst together with a base and acetonitrile, and you use this D-ketocarbonyl compound. And then what happened is that this is cleaved here, and this C3 unit, in essence, appears then is then coupled to this bromine here. And that actually turned out to work really, really well. So this gave us this intermediate here, where we have the carboxylic acid and this ketone now here. And then when you treat this with a bit of ammonia, it cyclizes around and gives us this intermediate. Now treatment with PuCl3 then gives us this great intermediate. And this is how we made all of this class compounds with this methyl group here. So in essence, what you can do now is oxidize this methyl sulfide. And this gives you a leaving group, and then you can, in a very sort of divergent way, introduce substituents here and here. And we also use this to make more than 1 kilogram of this drug. And again, this was done by a CRO, so by a different company. We don't have the equipment to do this sort of large upscaling. We don't have the expertise. But this CRO, this company, took our route forward, optimized it and tweaked it, and then made the drug in gram scale. Okay, I'll just leave it there. Just would like to acknowledge everyone who was involved in the MPS I project. It was a huge project. Particularly like to thank Spiro Leonardopoulos, who had the initial idea of exploring MPS1, my former colleague Julian Black, who's also used to be a chemistry team leader, and his team, they did a lot of the early ASA indole work, everyone else. And I would like to especially thank my members, thank the members of my team. Unfortunately, because of the situation, I don't have a recent photo of the team, and almost everyone who worked on MPS1 has now left. So huge credits really for most of the pyridopyrimidine work. So making this compound, this drug BOS 172722, goes to two postdocs in my lab, Hannah, who's now I think at UCL, and Paolo, who's now in Netherlands. So they're really Mr. and Mrs. pyridopyrimidine, and huge credit to them for making this drug. Saf was also involved and at the early stages, made one of the first compounds in this series. And Jack, who is still in my team, as you can see, probably wearing the ICR lab coat, also made significant contributions to this project. So this is really where I would like to leave it. I want to mention that we are currently recruiting both postdocs and PhD students in the Department of Chemistry. So we have 4 chemistry teams here. We are the Medicinal Chemistry Team 4, a very evocative name. But within the Department of Chemistry and also in my team, we are recruiting both postdocs in synthetic and medicinal chemistry, and PhD students in chemistry and chemical biology. And in my team, we're recruiting a PhD student to work on a computational chemistry project. Yeah, and you can find more information on the ICR webpage. And then I'd like to again leave it there and thank you for the invitation, and I'm happy to take any questions.
Oh, thanks. That was a really interesting talk. Quite interesting to see the sort of magic of methyl groups and what they can do to to change things. You said you wasn't a cancer expert, but I actually learned so much about cancer and what it is as a disease from this talk. I was really shocked by the relapse rates, actually. Those pictures of the skin cancer, the sort of before and after, and then after was, yeah, really shocking. Yeah, so we've got a few minutes for questions, so I'll just open it up to the floor. You can either post your questions in the chat, I've just put a little link, or if you want to raise your hand and I'll unmute people. It's probably the best thing. If anyone has any questions. Yeah, Chiara.
Thank you so much for your talk. It was really interesting, particularly like the end with the medicinal chemistry. I have a question about when you had your bulk drug, but you were trying to optimize it to make it more potent. How did you use the crystal structures to predict that?
And we used it in a variety of different ways, and a lot of that is actually published if you're interested, right? The citations are on the slides. So in this particular case, right, where we changed the scaffold, it's often referred to as rescaffolding, where you know, we go from one scaffold, central core, one chemotype to another one. We had crystal structure of the initial ASA indole series. We also had crystal structure of other compound series, and so we looked at them in the crystal structure, identified the critical interactions with the kinase, with the proteins, and sort of thought about ways how we can combine the best of all worlds, right? And how we can then translate this into a new molecule. In essence, what a crystal structure tells you is which parts of the molecule are important for activity, but also which parts of the molecule you can't really introduce any changes, and where you can introduce changes, and where these changes could then result in productive interactions and making the compound more potent. It's quite a visual task, right? It not necessarily involves computers, right? So you sit there, you look at these crystal structures. We have a big visualization suite here where you do that together. You scribble down compounds on a piece of paper, and then prioritize them different means, sometimes using computational docking, sometimes simply by, you know, how difficult or how easy it is to make these compounds.
We've got a few questions in the chat. Is there any agent that can help to reduce the drug resistance?
Yeah, I wish there was. And I went through the different concepts, but No, I mean, the only example I'm really aware of where you can essentially achieve cures, right, are some chemotherapies that have been around for a while, and these immunotherapies for some patients. But for most targeted cancer drugs, and that still is the case for the BRAF inhibitors, many other targeted therapies, lung cancer, EGFR inhibitors, right, you almost inevitably see resistance.
And a question from Tina Maria is, can inhibiting MPS1 be applied as a treatment of other types of cancers?
It can. For example, we're also testing the drugs, giving the drugs to ovarian cancers. And most cancer drugs will only work for a very, very specific patient population. And that again comes down to, in essence, that they explore a specific vulnerability of that cancer. For all other cancers, often the side effects will outweigh the beneficial effects of the drug, and that, in essence, is the challenge also of clinical testing, right? If you take a new experimental drug into clinical trials and you test it on a lot of patients who don't have this particular mutational type signature, then the drug simply won't be effective, right? And so you really need to design your clinical trials early on, so you have a good chance of seeing that your drugs work on these patients.
Okay, and then finally, the final question from Ahmad. How did the methyl group prevent metabolism?
That is a really good question. Let me see if I have a backup slide. I can see if I can find another presentation, if you don't mind. I actually quite like talking about that. I'll just see, how can I stop this? I really need to stop. I stop sharing for a moment, see if I can find something where I can explain that.
Yeah, it was quite interesting to see. There was 3 methyl groups, right? That was the difference between the one that
it's an interesting
metabolized too much.
Yeah. Let me just see if I can find that. It's a nice thing when you're at your desk, you can just dive back in.
Straight on.
Yeah. Let me see if I can share this. Yeah, so the problem we really had was, we had this drug, which was already part of this one slide overview, right, where it looked great for many ways, but then we have this huge turnover in essentially a model of a human liver, right? And there are ways how we can optimize this medicinal chemistry ways, and I come in a second to what's really happening there, right? There are different ways and strategies to solve this out, medicinal chemistry strategies. There's a lot published about that, but all of them didn't work for us, right? So it was quite an existential situation on our project, because we had already dropped once years, we had invested probably 3 years at this point, and probably a couple of £1 million, right? So we needed to find a way through that. And what really helped us was serendipity, right? And when we made more compounds, we made this one compound here, which had this methyl group, and actually following a different hypothesis, we thought this would make our compounds more selective. And it didn't turn out to do that at all. But what we found was that this compound was a lot more selective, so it had a lower clearance number in this model of human liver, right? But unfortunately, when you introduce this methyl group, you completely lose the activity. So there's about a 1,000-fold drop in activities. We couldn't explore this molecule. But working in parallel, we found another change here. When you swap this pyrazole with this triazole, right, that gives us a lot of potency. It gave us this compound, which was hugely potent. It's probably the most potent MPS1 inhibitor I know, even though quite a couple of companies worked on that. But it had the same problem as this one. It was highly turned over in models of human liver. But of course, then, we had a lot of potency here, we had stability here, and so the idea arose that we bring these two together. And this really led us then to that drug 172722. So then the question, what is really happening there, right? And the interesting thing is, what you do in these cases is you can use mass spectrometry to see when you add this to liver extracts, right, what is happening? Where are these compounds chewed up and metabolized, right? And the interesting thing was what we then observed in this experiment was that all these oxidations that you typically find in cytochromes occurred at this part of the molecule, right? So really quite far away from where we introduced the methyl group. And what we think is happening here, so this metabolization in human livers is done by P450 enzymes, cytochromes, and they're actually quite interesting in that they have two sides. They have a molecular recognition side, and they have a catalytic center, an active catalytic center that does the sort of oxidation, right? And they are connected by somewhat flexible linker. And what we're thinking here is that the methyl groups interferes with this molecular recognition, right? And hence we have an effect on metabolization in a different part of the molecule, even though the methyl group is far away. And primarily, route of metabolism we find here is no longer in this part of the molecule, but is now at this position, and it's much slower. Hence the better stability in human livers. A bit long-winded, but that's the story behind it. Yeah.
Oh, it's really interesting. Yeah, thank you so much again. So I'm gonna pass over to Miranda and Chiara. So what we're gonna do is kind of go into breakout rooms. So if Swen, if you want to hang around, and maybe people will have more questions for you then.
Yeah.
Cool. Okay. Bye, guys.
Bye.
Bye.